Go ahead and kick us off.
Great. Hi, everyone. Let me just share my share my screen. Can everyone see my screen? Great. Well, hello, everyone, and thanks for joining us. Every year, hundreds of scientific papers describe exciting new stem cell-derived models, organoids, co-cultures, new differentiation protocols, and increasingly sophisticated human in vitro systems. Yet only a handful of those assays become ones that pharmaceutical companies routinely use or that regulators are willing to consider when making drug development decisions. The question is why? The answer isn't simply better biology. It's productivity, reproducibility, standardization, and demonstrating that that model is fit for its intended purpose. Ultimately, the value of a stem cell-derived model isn't determined by how closely it resembles human tissue in the laboratory. It's determined by whether it generates reliable, interpretable data that can support a real decision. And now we're seeing some human stem cell-derived assays make exactly that transition from promising research models to technologies that influence drug discovery and safety assessment. I'd like to illustrate that using two examples I've been fortunate to work on. The first is the human iPSC cardiomyocyte MEA assay for assessing proarrhythmia risk, or in other words, it's the risk that a drug could trigger a dangerous abnormal heart rhythm. The second is the emerging human iPSC neural MEA assay for seizure liability. They're at different stages of maturity, but together they illustrate how a promising model can evolve into a trusted NAM. And by NAM, I mean a new approach methodology, broadly a technology or approach designed to answer a specific hazard or risk assessment question more efficiently than existing methods. The key is that a NAM should be fit for purpose, able to generate relevant interpretable evidence for the decision being made. For human safety assessment, that often means improving our ability to predict human biology and understand the mechanism. And when a NAM does that, it can also reduce reliance on traditional animal testing. For example, by identifying high-risk compounds earlier, reducing repeat studies, or supporting decisions where animal models are less relevant.